Hi, I'm Greg Petsko. I'm professor of neurology and neuroscience at Weill Cornell Medical College in New York City, and I'm also the director of the Appel Alzheimer's Disease Research Institute there. In this video, I want to talk to you about Parkinson's disease. I want to give you some information about how it starts and how it might be stopped. In an earlier video, I told you about the general problem of neurodegenerative diseases and the coming epidemic of such diseases. Parkinson's disease is the second most common neurologic disorder, affecting about 1.5 million people in the United States alone. I call it the body snatcher because Parkinson's disease leaves you cognitively intact until the very end, but takes away your ability to function physically. It's a hideous disorder. It takes about 20 years, 25 years to progress to fatality. As I said, the second most common of all the neurodegenerative diseases. Typically, the average age of onset is just about as old as I am. I'm 67. And about 1 in 7 people over the age of 80 develop Parkinson's disease. There are no treatments that modify the course of the disease or prevent it. There are treatments that deal with the symptoms, but while they're dealing with the symptoms, the disease goes merrily on. And like Alzheimer's disease, and in fact like Lou Gehrig's disease, 90% of all cases of Parkinson's disease are sporadic. They're not inherited, as far as we know, and they're idiopathic. We're not sure what causes them. 10%, however, are genetic, run in families with either an autosomal dominant or autosomal recessive inheritance, and those cases, as you'll see, have taught us quite a lot about what's going on with this disease. The disease was first identified by James Parkinson back in the 19th century, and it's a fascinating story, which he wrote about in his little essay on what he called the shaking palsy, but we now call Parkinson's disease after him. He was the first person to describe this as a regular disorder, although there are descriptions of what must be Parkinson's disease that go back thousands of years in Chinese scrolls. So it's been an affliction that's bedeviled the human race for a very long time. But Parkinson actually laid it out as a clear-cut clinical phenomenon. And he did this on the basis of only 5 patients, just 2 of whom he actually spoke to personally. The other 3 he observed while walking around the streets of London. If you want to know how to do a neurological diagnosis, read this monograph. It's brilliant. Michael J. Fox is one of the most famous victims of this disease. He has, as far as we know, the sporadic form of the disease and was unlucky enough to get it very young, in his late 30s. It is, in his case, a disease that he has used as a springboard to create a private foundation, the Michael J. Fox Foundation, which is one of the leading supporters of Parkinson's disease research in the world. The disease itself, well, it's got about the same incidence as thyroid cancer, and as I said, there are about 1.4 million people living with the disease at any given moment. And typically, it takes 20 to 25 years to progress to fatality. About, I would say, 1/4 to 1/3 of those patients actually become demented as well towards the end stages of the disease, and it's just an awful disorder. We have nothing that modifies the course of the disease. We have identified a number of genes that are responsible for some of the familial cases, and as I said, they've taught us a lot about this disorder, but we still don't have a way to stop it. And we're still not sure what causes the sporadic cases of the disease for which there are no obvious genetic causes. There are, however, risk factors. We know there are things that make it more likely you'll get Parkinson's disease. One of them would be head trauma from accidents, or in this case from boxing. Muhammad Ali is a famous sufferer of Parkinson's disease. Interestingly enough, infection of the brain by viruses. That also, in many cases, is a precursor to Parkinson's disease. The bird flu epidemic of 1918, 1920 was followed, in fact, by an epidemic of Parkinson's disease for many of the people who survived the flu. We know that certain mitochondrial poisons, things that damage the mitochondria, some pesticides, for example, will in fact give Parkinson's disease in animal models and will in fact give Parkinson's disease to people who work around these and ingest too much of them. High cholesterol is a risk factor for Parkinson's disease, not clear why. And people with lysosomal storage diseases, or people who are carriers for genetic lysosomal storage diseases, have a much higher incidence of Parkinson's disease. Again, the reasons why are not all that clear. But you might want to remember this list because we'll come back to it a little later. The reason you have the symptoms of Parkinson's disease, the tremors, the slowness of movement, the postural instability, all of the terrible things that could go wrong with you physically, is because you're losing a particular set of neurons rapidly during the course of the disease. Those neurons are the dopaminergic neurons of the substantia nigra, part of the midbrain that produces dopamine and that sends its axons into the nigrostriatal pathway, which controls the smoothness of movement. You can see, comparing the left, a normal person, with the Parkinson's patient on the right, that you've lost a lot of that black substance. That's the dopamine-producing neurons. And typically, in fact, about 70% of those neurons are gone when you present with symptoms of the disease. If you look at those neurons in the course of the disease, you find something in them that shouldn't be there. They're dense aggregates of misfolded protein. They're called Lewy bodies after the French physician who first identified them. And you can see on this slide that they stain predominantly for ubiquitin and for a protein called synuclein, which is a central nervous system specific protein about which not too much is known functionally. You'll notice also they're quite strikingly structured. The synuclein's on the outside and the ubiquitin's on the inside. Nobody knows why that's true. But if you analyze the Lewy bodies biochemically, and we and others have done this, you find that they contain predominantly alpha-synuclein. There's also a fragment of alpha-synuclein, and then there's ubiquitin, as I said, and a lot of other stuff in various amounts and more depending on how willing you are to look deep into the list of minor components. Here's synuclein. It's a 140 amino acid long protein. As I said, we don't know as much about it functionally as we should, but what we do know is that some of the mutations that give you Parkinson's disease occur in this protein, and those mutations are shown here in red. Those residues, mutated as you see here, guarantee that you'll get Parkinson's disease, and generally speaking, you tend to get it a bit early. In addition, there are rare families that have wild-type synuclein sequence, but that have duplications or triplications of the synuclein gene. So they just have a higher load of wild-type synuclein. And they get Parkinson's disease, and they get Parkinson's disease young. So these genetic factors strongly implicate synuclein, which is found in the aggregates after all, as a critical component for Parkinson's disease. Now, we did a lot of work a while back, summarized on this slide, that showed that under the right circumstances, you could prepare synuclein as a fairly stable, mostly alpha helical, tetrameric protein, whose NMR was that of a partially folded protein, unless you denatured it, in which case, of course, it became a disordered protein, and that this protein basically was incredibly stable over time, unless you denatured it, in which case it rapidly aggregated, that's the red curve there, and formed things that look like little Lewy bodies, filamentous amyloid aggregates of synuclein. And these experiments, which were published, raised a question that bothered us a lot. If fundamentally synuclein is not that aggregation prone unless you have mutants, I mean, if you put the Parkinson's causing mutants into synuclein, you lose a lot of the helical structure and you get rapid aggregation, as you can see here. Well, that makes sense, but remember, most people with Parkinson's disease don't have any mutations in synuclein. It's sporadic and idiopathic, yet they have synuclein Lewy bodies, aggregates of synuclein. So if synuclein is not that aggregation prone intrinsically, then how the heck does anybody ever get Parkinson's disease? That was the question we wanted to address. And it seemed to us that the way to tackle that problem might be to go back and look at those Lewy bodies again. Because remember, there's a fragment of synuclein that was found in the Lewy bodies. And that's in fact a very specific fragment here. And we wondered, what about that fragment? So we fished it out, we did mass spec, and we found that the fragment was produced by a cleavage after aspartic acid 121. It's where that red arrow is right over here. And what you can see is the last 19 residues of synuclein are cleaved off. That's that fragment. So we made that fragment. We just designed a gene for it, made the protein, looked at it. And it turns out that protein behaves very differently from full-length synuclein. It aggregates like crazy. And that got our attention. If the fragment's in the Lewy bodies, and if the fragment aggregates much better than full-length synuclein, then maybe fragment formation is a very early event in the disease. Maybe, in fact, it's the early event in the sporadic disease. It's what's triggering the aggregation of the full-length protein. Now, if that's true, then the question obviously is what's cutting synuclein at residue 121? What's the protease, the protein-cutting enzyme that's doing this job? Because proteases are great drug targets. We know how to inhibit proteases. Problem is, there are hundreds of proteases in the human genome, and many of them are essential genes. So it's very hard to get at what the right protease might be. And so to do that, we turned to a model organism. And the model organism we turned to is yeast. Now I know what you're thinking. You're thinking, wait a minute, yeast doesn't have a brain. This is true. But if you've been following the presidential primary season, you know that not every person has a brain. And so clearly brains are not essential. And in fact, although yeast doesn't have a brain, it's a great model organism if you're wondering what goes on in fundamental cellular processes. And Tiago Otero in Sue Lindquist's lab had shown 15 years ago that synuclein is toxic to yeast when you overexpress it. Kills the cell. And in fact, in doing so, it produces, as you can see on the left here, it produces these fantastic little Lewy body-like aggregates. Okay. And that suggests that there may be something going on in yeast that's somewhat similar to what's happening in a neuron. Well, Tiago and Sue went on to do a lot of fascinating genetic experiments with this system, but they didn't look biochemically at what was going on in those aggregates. So we thought this was a good opportunity to do that. We fished those aggregates out of yeast and we analyzed them, and guess what? They contain exactly the same fragment that you find in the Lewy bodies of people. Exactly the same fragment. Which means that yeast contains a proteolytic activity capable of cutting synuclein at the same place that it's cut in neurons. Well, the fascinating thing about yeast, of course, is there aren't 500 proteases in yeast, there are about 50. And you can do genetics in yeast really easily. So what we did was to design a screen in which we took these synuclein overexpressing yeast that would normally die, and we knocked out one by one the protease genes in yeast. And we asked the question, can we find a protease that if you get rid of it, synuclein no longer aggregates in yeast and the yeast can survive? No fragment, no aggregation, survival of the cell. Straightforward genetic screen. We screened 5,500 deletions of the yeast genome if you were to screen every yeast gene, but remember, we're only screening proteases. So we screened 50, and that's an easy thing to do. And it turns out there are two of them that when deleted actually rescue the cells from synuclein toxicity. One of them is YCA1, which is the only yeast homolog of a family of proteases called caspases, and the other is RIM13, which is the only yeast homolog of a family of proteases called calpanes. And in yeast, these proteases are connected, one activates the other, so we're probably looking at a single proteolytic event. And these are both cysteine proteases. It's a specific family of proteases here. Well, now we've simplified the problem enormously because in neurons, there are 29 caspases plus calpains. And that's a manageable number. So what we then set out to do was to knock them down one by one and to see if we could see the same thing we saw in yeast. Because in yeast, when we knocked down the protease, there was no longer a fragment and no longer any aggregation. And those are easy things to look for in neurons. So we used RNA interference to knock down each of the caspase and calpain genes in yeast. In neurons. And to find out whether we could see a phenotype relative to what we saw in yeast. We needed a model in neurons for Parkinson's disease. Mark Cookson provided one. It's a model in which normally, as you can see on the left, oxidative stress is not particularly toxic to neurons in culture unless you overexpress synuclein. That's the figure on the right, in which case at a particular threshold concentration of oxidative stress chemical, the cells rapidly die because they have synuclein. And this model had been used for quite a number of years, but nobody quite understood why you had to have both oxidative stress and synuclein. Synuclein on its own wasn't toxic. You needed a huge amount of oxidative stress without synuclein. What's the reason for this synergy? So we looked at this model and we found the reason is the fragment. It turns out the fragment appears, you can see there where the arrow is, exactly at the concentration of oxidative stress inducer, menadione, exactly at the concentration where the cells become toxic in the presence of synuclein, where the cells start to die. So fragment formation is directly linked to toxicity in this model. It's exactly what we need for our knockout experiments. So we took this model and one by one, we knocked out the caspases and calpains. And only one of them made a difference. And that one was caspase 1. You can see, for example, on the left, that when you knock down caspase 9, you still have the fragment, there where the red arrow is. Whereas when you knock down caspase 1, the fragment disappears. So it's caspase 1. And caspase 1 got our attention big time, because even though it has the same name as a cell death protease, caspase 1 is not a cell death protease. It is a cysteine protease. But what it is it's the major inflammatory protease of the human genome. Caspase 1 is linked to inflammation. And we did a quick set of experiments summarized on this slide in which we proved that in vitro, caspase 1 could cleave synuclein to make the fragment, that's the figure on the upper left, and that the fragment was cleaved at the right place, that's the mass spec on the lower left, and that if you added a pinch of caspase 1 to a stable preparation of synuclein, here, synuclein rapidly aggregated. So this looks like it has the characteristics of the protease we're looking for. Cleaves in the right place, the reduction of the protein to a smaller fragment produces an aggregation-prone fragment. Everything we wanted seems to be here. And caspase 1, because of its role in inflammation, has been studied for 30 years. There are good crystal structures. Here's one we did, but there are many that were done before. And even better, there are chemical inhibitors for caspase 1. People years ago were interested in inhibiting this enzyme for treatment of psoriasis, arthritis, other inflammatory diseases. Now, in fact, none of those drugs did a lot because inflammatory diseases involve more than just caspase 1. But they made a lot of inhibitors. And some of these inhibitors were even tested in people and were found to be safe. Unfortunately, almost none of them get into the brain. They don't have good blood-brain barrier penetrance. The best one is the one at the top here, which is VX765 from a company called Vertex Corporation. It's a prodrug that's converted here to the active drug inside the cell. That has some brain penetrance, but it's not great. We did, however, start to use it to see if it would work in neurons in our neuronal model of the disease. And in a dose-dependent manner, it restored survival of those neurons in the presence of synuclein plus menadione. Just what you'd expect if you were inhibiting caspase-1 and preventing the fragment formation. The neurons should survive just like the yeast survive, and they do. And so this is a chemical equivalent of knocking out the gene, and it works. If only we had a blood-brain barrier penetrant version of this inhibitor, we could immediately try it in animal models of Parkinson's disease. But at the moment, we're still trying to find such a compound. However, what I've shown you, I hope, is an explanation for at least some of the sporadic cases of Parkinson's disease. If inflammation triggers caspase-1 and caspase-1 cleaves synuclein to a aggregation-prone fragment, and that aggregation-prone fragment then produces various amyloid-like substances that are then toxic to the cell, you've got a reasonable model for how Parkinson's disease might start. It does raise the question, though, do we have any evidence that this really happens in Parkinson's disease? Now, the first thing I'd like to ask is, what activates caspase-1? You don't want to have an active protease running around in the cell. Well, the answer is caspase-1 is activated by a complex called the inflammasome. It's normally part of the inflammasome, and when the inflammasome, which is a sensor of inflammatory events, is activated, caspase-1 becomes activated. Okay, so what activates the inflammasome? Cholesterol deposits, viral infection, head trauma, mitochondrial poisons will do it, and lysosomal damage will activate the inflammasome. Does this look familiar to you? Yeah, this was the list I showed you earlier of the risk factors for sporadic Parkinson's disease. They happen to be the things that activate the inflammasome. Might be just a coincidence, but it fits beautifully with the model that we're talking about. And here's something else that fits. If you actually look in the brains of Parkinson's disease patients, caspase-1 is highly active in precisely the region, the substantia nigra, that is most vulnerable in the disease. You can see that there in the red bar in the middle. So I think we have a plausible hypothesis for how Parkinson's disease starts, that inflammatory events activate the inflammasome, that the inflammasome then produces active caspase-1, which cleaves synuclein, and so forth. If this is how Parkinson's disease starts in many cases, then it's also how Parkinson's disease might be stopped. And all we have to do in order to stop Parkinson's disease might be to find a blood-brain barrier penetrant form of caspase-1 inhibitor. And that's something we're actively pursuing in my lab right now. So we haven't quite gone yet from molecule to man, but I'm hoping we will. And we'll get a chance to test this hypothesis the way it has to be tested in animal and then eventually human disease. If we can show that, then we might eventually be able to provide someone with a disease-modifying treatment, not just a symptomatic treatment for Parkinson's disease. Now, they say no man is an island. It's particularly true in scientific research. This is the team that's been working on this problem. My colleague Dagmar Ringe at Brandeis, postdoc Quen Huang, who did many of the experiments I've shown you, together with postdoc Xulin Zhu, and our collaborators at Brandeis, at Bordeaux, and at Rush Medical School and NIH, who've helped us along the way with many of the things that we've had to do that we weren't familiar with. Thank you.